So what we'll be doing is looking at episodic memory, so a particular kind of memory that appears to depend on the hippocampus, and so we'll be focusing on that structure as well as on the behavioral characteristics of this kind of memory. Okay, so in terms of overview, kind of the learning objectives that we'll have in this module, we'll learn about the distinctions between different types of memory and their neural substrates. So this may be somewhat familiar to you, the distinction between declarative and non-declarative memory, and within declarative memory, what episodic memory is. We'll also talk a lot about structures in the medial temporal lobe and their roles in declarative memory. So we'll talk a lot about the anatomy of the medial temporal lobe structures. And we'll also, in the last lecture in a few weeks, we'll talk about how Alzheimer's disease affects these structures. So I'll kind of bring it into more of a clinical realm by talking about Alzheimer's disease particularly. Okay. So the medial temporal lobe memory system is here. You can see a view of the underside of the brain. You can see here are the temporal lobes. And if you look in the middle are the medial parts of the temporal lobe. Now, before the study of patient HM, who I think probably most of you are familiar with, from other classes, who actually died fairly recently, and now we've uncovered his identity. We can talk about him, named Henry Molaison. Before this particular patient, the idea was that memory was really very distributed throughout the brain. There weren't specific structures that subserved memory. And his case was really critical to the field of behavioral neuroscience in that because he had as you probably know, he had an excision of these structures in the medial temporal lobe, and as a consequence had a very severe deficit in memory, but it was fairly selective. So a lot of other cognitive functions were spared. So the study of this particular patient really opened up a whole new lines of research because it indicated that, well, memory is localized somewhat in the brain. It's not just the whole brain is doing it, which is sort of the view that people had before this particular patient HM. Also, before the study of patient HM, there wasn't really a very clear idea of what these structures in the medial temporal lobe did. I mean, the thought was, well, this is part of the limbic system, these structures here, including hippocampus, amygdala, and the cortex surrounding them. Maybe they have to do with emotion, certainly no real higher function. So it was a bit of a surprise that actually the lesion that HM had was actually caused this deficit in memory. And people often wonder, well, gee, why would they do such a severe surgery? I mean, certainly, as you probably know, HM had very severe epilepsy that was intractable to drug treatment. He had fallen off his bicycle when he was 9, got a head injury, and as often a consequence of head injury, he had very epilepsy, very frequent seizures. The idea was to remove the epileptic focus, to remove the place where the seizures were emanating from. And that was why the surgery was undertaken. However, at the time, they had no idea that it would cause this kind of very profound cognitive problem with memory. But the study of this particular case really allowed us to know so much. It just really was a huge advance in behavioral neuroscience. And HM, though he wasn't able to really learn new information in a lot of ways, the reports are that he was aware that he was being helpful by participating in experiments throughout his life. Okay, so just a little bit more information about the anatomy. Here's an MRI scan of patient HM, and you can see there's kind of dark spots here. This would be fluid. So this is a coronal section, so like this through the brain. So the front would be out here, and the back is into the wall. And the temporal lobes are right here, and the medial part of the temporal lobe, so here's medial into the middle, are are the parts that were taken out in HM. So the surgical approach was kind of to go in like this and remove all of this tissue. And in this scan, it's kind of black here because there's just fluid there, where most people would have a hippocampus and some cortical regions surrounding it. And if we look here at a section, this is through the center of the brain, you can see some of these structures that I'll be talking about. First of all, you can see in the hippocampus here, and surrounded by the parahippocampal gyrus and surrounded by cortex, the amygdala is here at the front of this medial temporal lobe system. So you can't, from the side of the brain, you can't see these structures because they're really medial. So this is a view from the center of the brain looking in, and the hippocampus is kind of covered up by this parahippocampal gyrus. If you could see through it, you would see the hippocampus down in here. So these structures, they're not visible unless you take a section through the brain because they're all covered up in cortex. Okay. So these are the structures we're going to be studying, the structures removed in HM, the structures that we're going to be studying in the lab. Okay, so what were the lessons learned from case HM? I made the claim that this was just a revolutionary case, that was probably the most important case in behavioral neuroscience. Well, why do I make that case? What was so important? So kind of the first lesson we learned from him is that the medial temporal lobe is critical for memory. That was not known in any way really before the study of his case. As I said, that was why surgeons felt that it could be justified to remove these structures. And so specifically, the hippocampus and the surrounding medial temporal lobe cortices. So when the lesion was done in hippocampus, they had to go through this cortical gyrus to get at it. So I think one maybe misconception or maybe one idea that you've gotten from previous classes is that the lesion in patient HM was the hippocampus alone. That was certainly taken out in case HM. But it's important to realize that through the surgical approach, also a lot of this cortex that's surrounding the hippocampus was taken out. Okay, so that, it turns out, was important also in the severity of his memory problem. Okay. And then I think from a psychologist's point of view, the importance of case HM was the idea that there's multiple forms of memory, that even though clearly HM had impaired memory, there were some things that he could still learn. And that the medial temporal lobe, though it had this role in memory, it was specific to a certain kind of memory, what we call declarative memory. Okay. So, and then other kinds of memory would be intact, things like skill learning, particularly like learning a motor skill, would be intact and not dependent on the medial temporal lobe. Okay, so as I mentioned, HM's lesion was not confined to the hippocampus. He received bilateral removal of the medial temporal lobe, which included the hippocampus, the amygdala, and the perirhinal and parahippocampal cortex. These are cortical regions in the parahippocampal gyrus that were removed because they went in through this surgical approach, and so they removed this cortex that's surrounding the amygdala and the hippocampus as well. Okay, so bringing you up to date, currently a major focus of research, I think everybody agrees with the idea that HM has a memory deficit. These structures are really important for memory, but if I bring you up to 2009 and what are the big research questions, I would say a major focus of research is whether these multiple structures that have been were removed in HM or damaged in other amnesic patients that have subsequently been studied. Focus of research is whether they have a similar or different role in declarative memory. Can we differentiate what each of their jobs is? Okay, so that's kind of through techniques like neuroimaging or looking at patients who have even more selective lesions. So that's kind of a current focus of research. And as I mentioned, we know now that other brain systems are important for what I'll call non-declarative memory. things that aren't your memory for facts and events, your conscious memory. I think skills and habits and forms of Pavlovian conditioning and priming, some of these other abilities seem to depend on other brain systems. So for instance, the striatum seems to be very important for skill learning. So we're not really going to focus on that too much in this module, but you're also probably aware that this is a kind of a selective kind of memory deficit that amnesic patients have, that other kinds of learning depends on other brain regions, that there are multiple memory systems. Okay, any questions about? Okay. So just to kind of illustrate, we're developing this idea of a taxonomy of learning with long-term memory kind of divided up into declarative and non-declarative memory. I think other terms maybe you guys have used are explicit and implicit memory. Is that one that's you use more than declarative, non-declarative? Okay. I consider those basically synonyms. So if you've used explicit, implicit, it's basically the same thing. I tend to use declarative, non-declarative, but they're really meaning the same things. So first, for non-declarative memory, as I mentioned, there's like skills that tend to depend on the striatum. Priming depends on neocortex, some kinds of classical conditioning like fear conditioning that you've probably heard a lot about, depends on amygdala. There's some kinds of learning that depend on cerebellum, of simple conditioned reflexes, non-associative learning, like habituation. So that's non-declarative memory. What we'll be focusing on really is declarative memory, which is really your conscious memory for facts and events. I think that's the simplest definition. So when you remember things from class that you use on a test, you're aware that you're remembering them. That would be an example of declarative memory. So just about all the stuff you learned in school. As well, if you remember what you had for breakfast this morning, so you're remembering a particular event, what you did last night, I could ask you, you're consciously aware you're remembering it. That's really your declarative memory. It's sort of what you typically think of when you say, oh, I remember that. Well, that means you're declaratively remembering. It's really what you typically think of as memory. Whereas non-declarative memory, it's more kind of emerges through performance. Like, maybe learning to ride a bicycle. You're not exactly aware of what you're doing, but you can show that you actually get better at it with practice. As well as an amnesic patient can get better at these things without actually remembering that he's been practicing it. Okay, and so the declarative memory depends on medial temporal lobe as well as some of the output structures through the diencephalon. Not really going to talk about those too much. We're really focusing on the medial temporal lobe. Okay, so what do we know just looking at the anatomy of these systems? So clearly after the discovery of case-HM, huge amount of effort gone into studying these structures, how they're interconnected. What insights can we gain just based on the anatomical connections? Well, these cortical regions that surround the hippocampus, so the parahippocampal cortex and the perirhinal cortex, which are both part of the parahippocampal gyrus, the parahippocampal meaning surrounding hippocampus, they receive input from many, many other cortical regions all through the brain. They tend to receive input that's very highly processed from association cortices typically. They're not getting information from, say, V1 or primary motor cortex, primary auditory cortex. They tend to get information, say, from frontal lobe, parietal lobe, from lateral temporal cortex, where the information has been very highly processed already. So that's just one thing to note, just based on the anatomical connections. It seems like it's getting a wide range of information, and information that is already highly processed and kind of association from a bunch of different senses. You don't really see direct inputs from primary sensory cortex. It's kind of high-level stuff coming in here and converging in here. From the perirhinal and parahippocampal cortex, projection is to the entorhinal cortex, which then projects to the dentate gyrus of the hippocampus through the perforant path. Is this a little bit familiar to some of you, or no? New? Okay. So it's basic kind of anatomy of the hippocampus. Entorhinal into part of the hippocampus called the dentate gyrus. And that pathway is the perforant path. And then output of the hippocampus is to part of it called the subiculum. That's where the output is. And that goes back to the entorhinal cortex. So you see it's a loop. Okay? And so that's basically the structure of it. You have this highly processed information kind of in its kind of convergence into these cortical regions. They project into the entorhinal, goes into the hippocampus, and then back out from the hippocampus to the entorhinal, then back to cortex. So it's this kind of loop with hippocampus on top is sort of how you can think of it. And then what do we know from looking at lesion patients? Well, if you have a very selective lesion of the hippocampus, so just the hippocampus is damaged, but those surrounding cortices are intact, you definitely have an amnesia. There's definitely something wrong with you, and you can clearly demonstrate a deficit. However, the really kind of profound amnesia, like the kind that is described in case HM, where you'll talk to him, you'll go out of the room for 30 seconds, you come back in, he doesn't recognize you at all. That level of really profound amnesia really only occurs when you have lesions of these surrounding cortical regions, like HM did. Selective hippocampal lesions don't produce that huge, massive, profound amnesia. The other thing that we know from studying patients is the amygdala is not necessary for declarative memory. In HM, the amygdala was actually removed bilaterally. It's right there in the medial temporal lobe. And so just looking at case HM, you might have thought that was also important for declarative memory. Subsequent study of patients that just have the amygdala removed suggests that they have other problems, clearly, having to do with fear processing and emotion. But their declarative memory can be normal. What they're missing is that kind of boost you get for very emotional memories, where you can remember those a little bit better than very neutral, boring memories. So they don't get that kind of emotional boost. But for neutral materials, they can be really normal. Okay. So I went over the anatomical circuit, but this slide shows it as a diagram. So, and I kind of described it as this hierarchy. So this slide shows that you have unimodal and polymodal association areas in frontal, temporal, and parietal lobes, feeding into these cortical regions in the medial temporal lobe, to entorhinal cortex, and then into hippocampus. A primary input from entorhinal cortex is to the dentate gyrus, although there are, as this shows, there are connections to all the different levels, and there are reciprocal connections. But for the main connection is from the bottom up to the top and then back down again. The amygdala certainly communicates with hippocampus. I mentioned how emotions certainly can make memory stronger, very emotional events, and that's probably through these connections with amygdala. But if you take the amygdala out, you still can have declarative memory. Amygdala patients can have pretty normal declarative memory. And down here, we just actually see these structures in a section through the primate brain. And here is this is the temporal lobe here with the parahippocampal gyrus. It's kind of this piece of cortex that's surrounding the hippocampus that's right in there. This part is the entorhinal cortex, and here's the output, which is the subiculum. So this is the dentate gyrus, CA3, and the CA1. These are just parts of the hippocampus. So here's the hippocampus here, subiculum, and this is all cortex. And this shows those connections. Here's the perforant path coming from the entorhinal cortex up into the dentate gyrus, the mossy fibers going up into CA3, the Schaefer collaterals coming into CA1, then output to the subiculum, and then back out to the entorhinal cortex. Okay, so that is the basic anatomy of the hippocampal system. Might sound a little familiar, it might be new to you, but those are the structures that we talk about. Now, in addition, there are a lot of other pathways, but these are really the main ones. Okay, so it's sort of like this circuit where the information comes up, and then it comes back down to be stored. So the hippocampus seems like it has this role in kind of processing very high-level information. Yeah?
Coordinate path going to or out?
It's going to. So it is coming from the entorhinal cortex up into the dentate gyrus. So here's the entorhinal cortex, and here it is, I guess, in this section. And it's a bunch of fibers that come right in here to the dentate gyrus. A lot of people study that pathway who do LTP. I know you probably have heard of long-term potentiation. That is a pathway that's often studied in LTP studies. Okay, so these structures are very important for declarative memory, but in this module we're really going to be focusing on distinctions within declarative memory, particularly episodic memory and how it differs from other kinds of declarative memory. Okay, so this main distinction we'll be focusing on is episodic versus non-episodic. I said that declarative memory is basically your memory for facts and events. Well, there is an idea that the memory for facts versus memory for events, that they have somewhat different psychological characteristics and thus perhaps different neural characteristics. So that's kind of the idea we're going to be exploring. So episodic memory would be your memory for an event. I said, some kind of event like, what did you have for breakfast this morning? When I ask you that and you try to remember back, you kind of can travel back in time, kind of see where your position is in space, what you're actually doing. You can kind of recreate the room you were in and tell me about that event. Other declarative memories you have would be non-episodic, like, what's the capital of France? Tell me what day your final is in a certain class. Tell me what the name of your professor was that you had last year in 101B or 115. I guess you guys would take 115. And so kind of knowledge about the world. I mean, you're certainly aware that you're remembering that. That would be an example of declarative memory. You're using medial temporal lobe, but I'd say that's not episodic. You don't actually remember where you learned the Paris is the capital of France, or your knowledge of what your friend's phone number is, but that's distinct from your memory for an event. Another kind of non-episodic memory would be your feelings of familiarity. And I get this a lot because I often teach these large classes, and I'll be like at Ralph's or shopping, and someone will come up and say, "Hi, Dr. Knowlton," and they kind of look familiar, and I don't want to be impolite, but I'd like, "Oh, hi, how you doing?" But I really, of course, don't know. Do I know this person from this class or that class? Or are they in my department? Or, from all the different places I could know them. So they look very familiar. I'm very sure, very confident I know this person. I'm actually remembering them. I recognize them. But I don't have any. It's just a real strong feeling of familiarity. I don't actually remember, oh yeah, I remember you were sitting in the front, that lecture I gave last week. Okay, so that would be this feelings of familiarity. I'd call that a kind of non-episodic memory, but it probably is depending on my medial temporal lobe to give me that memory of, this familiar person. Okay? Yeah, question?
When I'm taking my exam, I'm thinking about that place, this is where I learned it. Would that all of a sudden be like an episodic?
That I would say is episodic. So yeah, it's interesting. If you were, say, taking an exam, I would say there's often a blend of episodic and semantic memory. You might, a couple of items, you just sort of know them. Then there are a few others that you think, oh, I remember when the professor was saying that. I remember what he was wearing, where he was standing, where I was in the room. You actually have a memory for that. That, of course, isn't necessary to do well on the test to have an episodic memory. As a teacher, what I want to do is convey semantic information to you. I don't care if you remember the configuration of this room or the color of the slide or anything like that, those kind of incidental details. However, I think when you are studying information, you often do have a bit of an episodic memory that might kind of come along when you're remembering things. However, those will tend to fade with time. So my guess is information you learned a few years ago in school, hopefully you still are retaining some of that. But my guess is that the episodic information, unless it was very salient and powerful to you, has gone by the wayside. But within an individual class, often I think you would have memories specifically of when you were studying something and what you thought of, and that might come back to you on a test. Any other questions? Okay. So episodic memory is characterized by occurring in a specific time and place. It's memory for an event. Anytime there's an episodic memory, I would be able to, or you would be able to tell me the actual place it occurred and the time it occurred. It happened at a particular point in time. And like Paris is the capital of France, when does that happen? Well, it's always true. I mean, didn't happen at a particular time, whereas, oh, yeah, the movie I saw last night going into the theater. I mean, that happened you might have a memory of that, be able to replay it in your mind, and that happened at a particular time and a spatial location. Okay, so that's kind of one key difference. Now, according to one view, the hippocampus itself plays a specific role in episodic memory. That when I ask you, okay, tell me what you had for breakfast yesterday, describe that event. You can describe sitting in your kitchen, your roommate's there, you had the last of the cornflakes, and you remember standing up and getting the milk. You can describe that event in detail. And a view is that you're really using your hippocampus when you're retrieving that. Whereas surrounding cortical regions are adequate to give you feelings of familiarity. So when I see somebody in Ralph's that I'm really sure I know, but I can't really place them, that I don't need the hippocampus for that kind of memory. But the surrounding cortical regions are adequate to give me that feeling of familiarity. Okay. So that's kind of one view of how hippocampus and surrounding cortex might have different roles. Okay, and here are some examples when people think of the big episodic memories of their life, like birth of their child or their graduation or their wedding. We often, if you're thinking about your graduation, you might remember a lot of little incidental details about it, like where you were sitting and how you felt, and you kind of replay it in your mind, that event, not just, I know I graduated on June 14th, 2004. That you might know that as a fact, but you actually kind of can relive it, these sort of big events. In the laboratory, we tend to not study these big life episodic events, but littler ones. Okay. So the episodic semantic distinction. So just to kind of go over the characteristics again, let me start actually with episodic memory, memory for events, memories tied to a specific time and place. The memory is from your own perspective. So when I ask you about your breakfast, you kind of can, when you're replaying it in your mind, it's sort of like from your own mind's eye, right? You're like, okay, I remember I was sitting here and looking at that wall. And you can often remember incidental details. So if I really try to say, tell me everything you remember about this breakfast event this morning, you can maybe tell what people were wearing or something you noticed. You could maybe describe the kitchen, these kind of incidental details that just happen to be there about the context, but they're not key to the breakfast experience itself. And it appears to specifically depend on the hippocampus for learning and for retrieval, actually, too. So let's contrast that with semantic memory, like your memory for facts, like the facts that you learn in class, for instance, including this one. They can be easily disconnected from the moment of learning. You might learn a fact in an episode, and the episode might stick around, but it doesn't need to. You can know that Paris is the capital of France just as strongly if you don't remember the moment you learned it as if you do. And they tend to be shared across people. We can all know these facts. We can all know facts about the world, people's names, and things that are true. So that's contrasted from episodic memory, where my episodic memories are, in fact, different from yours. I mean, they're not shared. Even if, like, example of a wedding, even the bride and groom have different episodic memories of that event because they had different perspectives. They're in different bodies. They're standing in different places. So they experience things differently. Whereas semantic memory is shared by, you say, all people in the culture basically know most of the same facts. And everyone in this class is learning some aspect of, hopefully, some large percentage of the facts that we're giving you. And this semantic learning appears to depend on the medial temporal lobe. So it's not like amnesic patients can learn facts normally. They still would have trouble, say, in a class like this, learning all these facts about the dentate gyrus and whatever. But it doesn't seem to depend on the hippocampus alone, that those surrounding cortices can kind of do the job fairly okay. So patients who, say, have selective hippocampal damage, they can sort of acquire facts about the world. Okay. So as I said, they can acquire facts about the world. One example is this case of a patient named John. He was born with a fairly severe hypoxic episode at birth, so perinatal complications that caused basically a lot of the cells in the CA1 layer of his hippocampus to die because they were deprived of oxygen. So he had a fairly normal brain, except a fairly specific loss of the hippocampus itself didn't work. So he grew up having very poor episodic memory. So you would talk to John. He'd say, well, where did I just put your keys? He couldn't remember that particular event. Where did you park your car today? He couldn't really describe, well, what did you have for breakfast today? I don't remember. But he was able to learn facts about the world. He did go to school. He had a normal IQ in terms of just knowing about the world. You could talk to him, and he certainly seemed to be aware of things that were going on and current events, and his vocabulary was normal. So even though this happened before he learned language, he learned the English language pretty normally, suggesting that maybe these structures that weren't compromised in his medial temporal lobe helped him do that kind of episodic memory. Now this is different, that kind of semantic memory. Different than, say, patient HM who had this much bigger lesion. He obviously had lost episodic memory. However, he really didn't learn new vocabulary words or new facts about the world after the 50s. Maybe a little bit, but really not normal. He really didn't, all these vocabulary words that are more modern, like website or jacuzzi, he never learned what those were. Okay, and also, who's president now, he would, keep thinking about Eisenhower. He wouldn't update facts, the way that John was pretty normal in this way. And there's also neuroimaging evidence that suggests that when you actually recollect information, so actually remember the moment you saw something, sort of go back to the episode that you saw something, you get hippocampal activation. When something just looks familiar, you tend not to. Also in paradigms of associative recognition, where you have a bunch of items paired together, and sometimes you remember, oh yeah, those two were paired together, or remember the moment I saw that, versus when you have those two items, they're not paired together, they just look familiar, but you can't actually remember the moment you saw them, you don't get hippocampal activation. So how do we measure episodic memory in the lab? So we'd like to be able to distinguish between these kinds of memory. And this is a task that you'll be using in the lab these next few weeks. One of them is called the Remember No task, where we're basically asking subjects to introspect about their memory. We give them a bunch of words, and then later we ask them, okay, for all these words you recognize, tell me, do you remember the moment you saw this word during the study phase? Can you kind of go back in time and relive it? Or is it just familiar? Are you just pretty sure it was on there, on the list somewhere, but you don't actually relive the moment you saw it? So that's the task we'll be using in the lab. Another way to do it is to actually ask subjects about source memory. We can say, okay, do you recognize this word? Okay, do you remember the background color? Do you remember the font it was in? Do you remember some incidental details? Because remember I told you how episodic memory tends to be accompanied by these sort of, you kind of see it in your mind's eye, so you see kind of the color and how it looked on the screen and things like that. Another paradigm that's used is associative recognition. When pairs of items are recombined at test, you can recognize the event as a new combination. So if I give you a bunch of pairs of words like window, reason, and then at test I might recombine window with an element of another pair like window, candle, and you can say, oh, well, those were paired differently. That suggests you actually remember the moment of seeing it. If you're just kind of going by familiarity, both window and candle are equally familiar. So you'll say, yeah, that was on there. But you won't get that they were actually recombined. So that's another way people have tried in the laboratory to look at episodic memory versus just sort of a feeling of familiarity. Okay. Here's an example using the Remember No task of some work that I did a few years ago where we were looking at the hippocampus and its activation when subjects were recognizing items, and when they gave an R, meaning they actually recollected the moment the item was shown. So this solid red line is for the activation here in the hippocampus. This is looking down onto the brain from the top. And if you look at the hippocampus, you can see that the only time it's really activated versus times where you don't recognize items is when the subject actually remembers the moment it was shown. So when they say, yeah, I recollect that item, that word snowman, I remember that it was on the list because when you showed it, I remember I thought about Christmas break. Or versus this black line here is down with the unrecognized items. That's when, yeah, you're correct that you actually do recognize the item, but just rings a bell, looks familiar. Cowboy, that looks like a familiar word. I bet it was on there, but I actually can't relive the moment you showed it to me. It just rings a bell. That doesn't seem to activate the hippocampus, even though you're correctly recognizing it. You can be quite confident. Okay. So there are arguments for the idea, against the idea that hippocampus has a specific role in episodic memory. So in this module, these are kind of the more cutting-edge questions that people are asking. I'm trying to, we all would agree declarative memory depends on medial temporal lobe structures, but it's still kind of up in the air, still controversial as to whether hippocampus really has this kind of specific role. And it is a little bit difficult to get at this. I mean, one real problem is that episodic memory, by definition, only takes place once. Okay? For semantic memory, you could have lots and lots of study trials. The reason that Paris is the capital of France, you've heard it 50,000 times, and just 3 times probably in this lecture alone. Whereas when I talk about an episodic memory, like what you had for breakfast this morning, that is a unique event in your life. It's only going to happen once, compared to, say, semantic memory, which you can really rehearse. So you have multiple training trials. And for familiarity, you could say, well, that's just sort of a weaker memory. Maybe these aren't two real different kinds of memory. It's just that the sense of familiarity and semantic memory, that's just, weaker memory. It's easier to kind of learn that. And the really hard memory is the episodic memory because it's only, you only get one shot at it. So maybe it's not that the hippocampus is special for episodic memory. It's that the hippocampus is just really good at the very hard stuff. And the other cortical regions can kind of take care of the easier stuff. So that's sort of another way to think about the data. And, well, what about the imaging results? Well, if the difference between episodic memory and semantic memory is more quantitative, it's not like, oh, the hippocampus is just doing this. It's sort of like hippocampus is good for hard memory so that when you have a recollection, that's like more memory. And if you just something looks familiar, it's less compared to having no memory. And maybe the hippocampal, the fMRI signal just isn't good at distinguishing those fine gradations, and you can only see a significant difference in the bold signal that's measured by fMRI in episodic versus the other kinds of memory, that it just isn't sense enough, that there may actually be a difference where there's a little bit of hippocampal activation for the just feelings of familiarity, just can't see it with limitations of the technique. So those ideas are also out there as well, currently. Okay, so then kind of the other view, the hippocampus has a specific role in episodic memory, and the other view is that the medial temporal lobe has this general role in declarative memory, and the hippocampus is critical for the most difficult memories, which, as I said, happen to be episodic. In either case, hippocampus is important for episodic memory. No one disputes that. The question is more, does it have a specialized role or not? That's sort of the state of the field. Is this specifically what it does, or is it just good for really hard memories, of which episodic is a really good example? Okay, so this is the last thing I want to do is tell you what you'll be doing in the next few weeks in this module. It's really two parts. One, we're going to be doing a behavioral study where we're going to use the remember no task to assess episodic memory in you guys. So we'll give you a series of words next week, and then the following week we'll test your memory for that. And on the memory test, the recognition test, you'll be asked for each word you recognize whether you actually remember the moment you saw it or if it just looks familiar. Okay, so we'll use this remember no task. We'll be going over that in a lot of detail. And the questions that we're interested in with this is whether episodic memory is associated with memory for more details about the layout of the screen, to really test the idea that when you actually remember the moment something is seen, when you actually have an episodic memory for it, you have memory for incidental details. We'll also look to see if episodic memory is associated with genetic risk for Alzheimer's disease. So what we'll also ask you, this is all done confidentially, is the number of relatives you have with dementia. And we'll see if there's any relationship between this proxy for genetic risk for Alzheimer's disease and your actual episodic memory abilities. The other study that we're going to be doing is a neuroanatomical study. So we're going to look at hippocampal volumes. We're going to actually measure them in MRI scans. And the questions that we'll be asking are there differences in hippocampal volume across young adulthood? So we know there's differences in aged subjects from young. But we're going to be looking at subjects in their 20s and 30s to see if we can actually see a difference within this younger group. We'll look at sex differences and then look at laterality differences. Are there differences for older and younger subjects in left versus right hippocampus, and also sex differences there. So I will see you next week in lab.